Good day, everyone. I'd like to give a presentation about our team published on iMeta. The study title is Host Selection Tendency of Key Microbiota in Arid Desert Lichen Crusts. Deserts represent arid environments with poor soil nutrients, where vascular plants have low coverage, but biological soil crusts, BSCs, have unique advantages. BSCs composed of cyanobacteria, algae, lichens, microfungi, and bryophytes, among which lichens play more crucial roles such as promoting nutrient cycling and improving soil physical stability. During the development and succession of BSCs, the predominant organisms are present in the order of algae, lichen, and moss. Lichen lobes, anchored by rhizines, bind sand particles, and drought-tolerant terricolous lichens, could play a significant role in the development of biological sand control. Lichens are symbiotic associations of microbiont with photobiont. They also include associated microorganisms, such as bacteria and fungi. Lichen-associated bacteria may be involved in many hosts' life activities and community. However, the potential assembly mechanisms of lichen's microbiota and how the lichen symbiont maintained its homeostasis are poorly known. There are also gaps in the community characteristics of the terricolous lichen microbiota, co-occurrence network structures, and potential drivers. Addressing these questions, we selected the lichen genus Endocarpon, considering its wide distribution in arid deserts and remarkable carbon and sand fixation potential. We collected two types of endocarpon crusts in the deserts of China and subdivided them into 4 distinct strata for the first time. We then amplified the 16S rDNA V4 region from and analyzed bacteria composition. The next is results. The biocrust stratum had the most significant effect on the Shannon and alpha index of bacterial community, followed by sampling sites and lichen species. The alpha diversity indexes exhibited a stepwise declining trend from decreasing trend in a stepwise manner from S to CS, and L to R. The CP-CoA and Permanova test results revealed that the most community variation was explained by the different strata, and no significant differences between the two endocarpon species. The taxonomy of major bacterial compositions found that oligotrophic taxa were more predominant in soil samples. In contrast, the relative abundance of saprotrophic taxa was higher in lichen samples. CCA analysis showed that the collection sites map and MAT significantly influenced bacterial community composition. DPA analysis revealed that strata explained most of the variability in the endocarpon bacterial community, greater than environmental factors and space distance. Source tracking analysis suggested that species filtration on the bacterial community gradually increased from the bottom to the top of the stratum, and lichen-associated bacterial communities were primarily derived from crust soil. A null model analysis of community assembly revealed that stochastic processes, mainly driven by homogenizing dispersal, exerted a greater influence on bacterial community assembly within lichen samples. In contrast, deterministic processes, predominantly governed by heterogeneous selection, played a more prominent role in soil samples. We constructed a co-occurrence network between the 4 strata separately. In summary, stratum affects bacterial network topology and keystone species. The S network showed significantly higher robustness, L network had the second highest connectedness, centrality, and complexity. The study provided key information to better understand the homeostasis maintenance mechanism of the lichen symbiont and community assembly of desert lichen crust. Thank you for your listening.